Welcome back to my YouTube channel Education Science. Today we'll be discussions by transduction in bacteria. Paper first microbiology. Content introductions. Discovered definitions in transduction, bacteriophages, transduction phases, first virulent phages, second temperate phages. Mechanism of transduction, types of transduction, general transduction, specialized transduction, application transduction in bacteria, introduction. Bacteria recombination is a type of genetic recombination in bacteria characterized by DNA transfer from one organism called donor to another organism as recipient. This process occurs in three main ways. Transformation, the uptake of exogenous DNA from the surrounding environment. Transduction, the virus-mediated transfer of DNA between bacteria. Conjugation, the transfer of DNA from one bacterium to another live cell. Contact. Discovered. Transduction was first discovered in 1952 by Joshua Lederberg and Norton Gender. Example: Salmonella typhimurium. Joshua Lederberg, Norton Gender. This is scientist diagram. Definition of transduction: the transfer of genetic material from one bacterial cell to another bacteria cell by a bacteriophage is called transduction. Bacteriophages. Bacteriophage is a virus that infects a bacterial cell. First described by Frederick Toward in 1950 and studied by D. Hurley 1970. Example: Escherichia coli. Example: Escherichia coli, Salmonella, Pseudomonas, Staphylococcus, etc. Transduction phases. A phage virus that transfer genetic material, DNA from one bacterial cell to another, is called transducing phases. Structures of bacteriophages: head, DNA, collar, sheath, middle part, sheath, tail, fiber, base, plates, pin, etc. This is diagram bacteriophages. Virulent phages. Virulent phages are type of bacteriophages that replicate only with the lytic cycle, do not from prophase stage. Lytic cycle, generalized transduction. Temperate phages. Temperate phages are bacteriophages that replicate using both lytic and lysogenic cycle. From prophase stage, both lytic and lysogenic cycle. Specialized transduction. This is diagram lytic cycle and lysogenic cycle. Example of bacteria. Transduction has been found to occur in variety of prokaryotes, including creatine species of bacteria, Escherichia coli, Pseudomonas, Staphylococcus, Xanthobacterium, Salmonella. The bacteriophages containing the bacterial DNA is called transduction. Types of transduction: transduction, generally two types. First, trans generalized transduction, specialized transduction. Second, generalized transduction, DNA fragments is transferred from one bacterium to another bacterium. A lytic bacteriophage is now carrying donor bacteria DNA due to an error in maturation during the lytic cycle. This is process diagram. Lytic cycle. First, A phages infect the donor bacterial cells. Phages proteins coats bacterial chromosomes. Second, phages replication and the bacterial chromosomes is degraded into pieces. Third, phages assembly. Occasionally, during phages, assembly pieces of bacterial DNA are packaged in a phages, copies the donor cells, lyses, and release phages particles containing bacterial DNA. Fourth, a phages carrying bacterial DNA infected another cells and delivers donor cells DNA. Fifth, integrations of donor DNA and recombinations can occur producing a recombinant cells. Donor bacterial DNA, recipient's bacterial DNA. This is structures lytic cycles. This is process transduction. Generalized transduction and specialized transduction. Generalized transduction, bacterial host cells, donor cells infected by phages. Second, host cell DNA is broken down into smaller pieces, proteins, and phages. DNA is also synthesized. Third step, bacterial host DNA is packaged in one of the viral copies that are released through lysis of the bacterial cell. Fourth, transducing phages with host DNA infect new recipient cell. Recombination also occur here. Fifth steps, recombinant new cell has a mixture of the donor DNA first and on of its own DNA. Its genotype is different. New recombinant cells. This is process of specialized transductions. First, prophase DNA integrated into the bacterial DNA. Prophase second step, prophase DNA cuts incorrectly and exchanges its DNA to, to that of bacterial host cell. Beginning of extension after complete exhalation. Third steps, phage copies contains bacterial host DNA, which is a transfer to a new bacterial recipient cell. Fourth step, transduced phages in transferred to a new cell and recombination occur. Recipient DNA. Fifth steps, the recombinant cell DNA has a different genotypes which is different from the donor or recipient cell genotypes. Generalized transduction step. Alytic bacteriophages adsorb to a susceptible bacterium. Second, the bacteriophages genome enters the bacterium. The genome direct the bacterium is metabolic machinery to manufacture bacteriophages component and enzymes. Occasionally, a bacteriophages or cap is assembly around in the fragments of donor bacterium. Nucleoid, nucleoid instate of a phage's genome by mistake. Fourth step, the bacteriophages are released. The bacteriophages carrying the donor bacterium DNA absorbed to a recipient bacteria. Fifth step, the bacteriophages insert the donor bacteria DNA it is carrying into the recipient bacterium. Seventh step, the donor bacteria DNA is exchanged from for some of the recipient's DNA. This is called the structures and process of generalized transductions. This is bacteriophages and the plasmids, bacteria, nucleoids, viral genomes, injects, integrates, penetrations, genomes, viral genomes, and phages carrying bacterial DNA. Fragments of the bacterians in the nucleoids of in the plasmids. Bacteriophages carrying donor bacterium DNA. Lytic bacterium or normal bacteriophages. This is diagram. See now. This is donor bacterial DNA. Donor bacterial DNA and inject the plasmids and donor bacterial DNA. Recipients bacteriophages nucleoids. This is process of specialized transductions. A DNA fragment is transferred from one bacterium to another by a temperate bacteriophages that is now carrying donor bacteria. DNA due to error A spontaneous inductions during the lysogenic life cycle. In specialized transductions, the phages insert its genome at the specific sites. Example of lambda is a good example of a specialized transduction phages. Steps involved in specialized transduction. First, adapted bacteriophages absorb to a susceptible bacterium and inject its genome. The bacteriophages inserted its genome into the bacterium a nucleoids to become prophase. During spontaneous induction, a small pieces of the donor bacterium's DNA is picked off as a part of the phage's genome in place of some of as the bacteriophages replicates. This is process of diagram specialized transduction. Golgi and viral genes or biogenes. Viral genes, this is bacteriophages genome. This is process and diagram. See now. The segments of bacterial DNA replicates a parts of the phages genome. Very phages now carrying that segments of bacterial DNA. The phages adsorb to a recipient's bacterium and injects its genome. The phage's genome carrying the donor bacterial DNA insert into the recipient's bacterium. Applications: transductions bacteria. It transfers genetic material from one bacterial cells to another and alter the genetic characteristic. It provides resistance to antibiotic drugs, help in the corrections of genetic disease by a direct modifications of genetic errors. It is a common tool used by molecular biologists to stably introduce a foreign gene into a host cell's genomes. Thank you.